Hi and Assalamualaikum warahmatullahi wabarakatuh. My name is Nashim Najis, and for today, I would like to talk about my thesis proposal, which is the title is Transfer Factors of Heavy Metal in Gills, Bones, and Tissue, which is in fish species located at coastal area around Selangor. Based in this slide, it consists of the presentation outlines, which consists of for the chapter 1 is introduction, chapter 2 is literature review, chapter 3 is methodology, and the last one is the references. For my thesis proposal, my objective is the first one is to determine the concentration of heavy metals in the gills, bones, and tissues of fish species in coastal areas around Selangor. And my second objective to investigate the relationship between the factors of fish species, size of organisms, and also environmental conditions to understand the influence on the bioaccumulations of heavy metals and other pollutants in the coastal ecosystem. And for the third objective is to determine the bioaccumulation of various heavy metals such as zinc, lead, and also cadmium that will contribute in the fish gills, bones, and also tissues of selected fish species inhabiting coastal areas around Selangor. And next slide is for the research background, which is my research background is focusing on the research will provide insight into the extent of heavy metals, contamination of fish species found in the coastal area around Selangor. And the second one is the study will provide valuable insights into the levels of heavy metal contamination in fish species which is inhabiting the coastal area around Selangor, and also to analyze the accumulation patterns of heavy metals in gills, bones, and also muscle tissues of the fish, which is we use two types of fish. And also the last one is to investigate the transfer factors of heavy metals that will occur in fish based on two types of different species of fish. And next, we go to the problem statement, which is the first problem statement that occurs in my proposal thesis is a lack of comprehensive data on the transfer factors of acid metals within different fish tissues. And for the second one is the potential health effect of transcendental contamination in marine fish and seafood, especially for frequent marine species consumers, pose concerns despite the health benefits associated with consuming these marine species. And for the third one is the increasing susceptibility of coastal regions to pollutions from anthropogenic activities, which is results in the accumulations of the heavy metals in the marine environment will be occurs. Next, we go to the this slide, which is consists of significance of study, which is the first one is the identifications of contamination, sources and bioavailability, and ecological impact. And for the second one is the understanding the levels of heavy metals accumulation in different fish tissues such as gills, muscle tissues, and also bones that contributes to evaluating the risk posed by heavy metals pollution into aquatic organisms, the food web, and the overall health of the coastal environments. And for the third one is the significance of this study lies in assessing the transfer factors of heavy metals in fish tissues as it provides crucial information on the potential health risks. The coastal area around Selangor are vital ecosystem for the marine life and also local ecosystems or local resources. Human activities such as industrializations and urban development are causing heavy metal pollution in the region. And this pollution has harmed the marine ecosystem and also human health, which is especially in estuaries and also has impacting the fish. And the toxic heavy metals like cadmium, zinc, and also lead can are being introduced through the human activities, and this contamination has affects the fish species and also marine species which has contributed to the presence of heavy metal accumulation in Kuala Selangor, for instance, and these pollutants has worsened the issues and had endangering the marine life and sustainability. And the transfer factor is known as the measure of the efficiency with which heavy metal is transferred from water to a fish tissue. And the transfer factor of a particular heavy metal will vary depending on the species of the fish and also the type of tissues and the environmental conditions. And the contamination of heavy metals to marine life also give a bad effects to the human health, such as because of human also consume the water, and it will give a toxics to will give a disease or toxics to the human body. Based on this slide, it consists of the types of heavy metals such as zinc, nickel, and also platinum. But it consists more than that, such as it also consists of zinc, lead, and other types of heavy metals that can appear to contribute give a bad effects to the ecosystems. And the image below is consists of sources of heavy metals, which is the sources of heavy metals can comes from the industrializations, mining, and also agriculture runoff. And now I would like to give information about the gills, bones, and also tissues that occurs in the fish species. Okay, for the gills, based on these pictures, for which is the upper one is we can see it. It consists of gills, which is the fish will absorb the heavy metals through their gills and while breathing, and then this metal can travel to the body parts through the bloodstreams. And the second picture consists of bones of the fish, which the fish bones plays a vital role in accumulating the distribution of heavy metals in fish. Exposure to heavy metal leads to their build up in the bones over time and through absorption from water or through the food chains. And for the last image that we can see from the above, it consists of muscle tissues of fish, which is the fish will absorb the heavy metals, accumulating them in the tissues like liver, kidneys, and also muscles, which is consuming such fish can pose to health risks, and monitoring and reducing heavy metals levels in coastal areas are very essential to do. Right now, this slide shows the process of bioaccumulation, which is occurs in the marine species of fish. Okay, here's the bioaccumulation of heavy metal in fish. As we know, bioaccumulation is when heavy metals enter the food web through water or organism, which has the accumulating in edible fish. Metals like lead, mercury, cadmium, and also arsenic, which is come from the human activities, can build up in fish over time. The fish will absorb these metals and then can reach to the human through the food chain and can causing the health effects. And the level of heavy metal accumulations in fish is influenced by variation factors such as the environmental concentration, fish species, and also the feeding habits. And different fish tissues may accumulate varying levels of heavy metals, with liver and kidneys usually having higher concentration than muscle tissues. And based on this figure one that we can look from this slide is which is it illustrates the bioaccumulating for heavy metals, which is the industrial has released metals which causes the small organism to absorb this, and then causing the metal concentration to rise up the food chain. So bigger organisms struggle to eliminate the metals as effectively. This slide shows the calculations of the bioconcentration factors of heavy metals in the soft tissue in fish. And as we know, the bioconcentration factors are determined by comparing the metal concentration in the fish tissues, and it are being measured in the forms of milligrams per kilogram. This slide shows the mechanism of the bioaccumulation process, which is the first process is the uptake, which is the marine organism can uptake the contaminants from the environment through the direct exposure to polluted water, sediment, and by consumes contaminated foods. And for the second process is the biotransformation. As we know, contaminants taken up can undergo biotransformation and converts into different chemical forms, and this process may lead to the creation of more toxic compounds. And for the third process is the storage, which is the following uptake and biotransformations. The contaminants are stored in the organism's tissues, and the process is known as the biomodification, which the bioaccumulation can worsen through the biomodifications, where certain contaminants becomes more concentrated and as they move up to the food chains. This shows the effects of the metal consumption. As we know, it can contribute to the cancer disease, birth defects, immune system dysfunctions, skin lesions, nervous system disorders, and last but not least is gastrostosis and also kidney dysfunctions. Based on this slide, shows the previous studies of the heavy metals that contribute to the effects of the marine ecosystem or marine life, such as there are two types of different species, which is known as Thalassianus, and also the second one is Chebelengeri. And this is the based on this shows the table of the concentration of heavy metals in Selangor that has contributed to give metal levels to the different types of fish. And also the concentrations are expressed in micrograms of metal per gram of dry fish. In general, the permissible limits of heavy metals allowed in fish muscles from both sides were lower than the permissible safety levels for human consumptions. And also, based on this result, indicated some sources of heavy metals uh, contaminations are present in the east side of the Peninsula Malaysia, despite relatively lower human activities. And the distribution of heavy metals within the fish tissues is a crucial process that occurs when they meet heavy metals in their aquatic habitat. This slide shows the methodology of the point of location for my sampling, which is located at the Kuala Selangor and also Kapar. This slide shows the fish species in coastal areas that consist of Ethalacinus and also Jebelengeri, which is can be seen a lot in the at the Kuala Selangor. For the methodology, the study area that will be conducted uh, at the Kapar and also Kuala Selangor, which is the area of the Selangor, and also for the procedure will be doing by between August until December 2023, and all the lab sessions will be doing at the UITM Applied Science Laboratory. And also for the sample collections is the water samples will be collected from 10 cm, which is below the water surface, by using the water sampler. And also um, we will choose the fish species, which consists of two types of fish species, which is Atarsanas and Jebelengeri. And also there the 9 samples of fish samples will be purchased, and all the collected samples will be kept in a refrigerator at a temperature which is below 4 degrees Celsius, in order to reduce the activities of the metabolisms of the organism in the water. This is the sample preparation, which is the sample preparation is also important because in order to before doing the lab samples, or before doing the lab sessions, because there are a lot of methods will be used, such as weight digestion method, which will be used in order to prepare the samples of the fish species, and also by using the analytical methods for atomic absorption spectroscopy with some modifications. And the fish sample will be put in a plastic tray and will be stored at a room temperature. And for the length of the fish sample will be recorded. And this is the type of species that I have mentioned before, Atlantis and also Jabalingari. And the land that that will be take notes to do, and more. Next is the sample analysis. The analysis of metals in water sample and fish tissue will be carried out. And for fish tissue, the analysis will be carried out using the Optima DV optical emission spectroscopy equipment. And the result from both of the analysis will be expressed in the unit of micrograms. And also for this is the data analysis, which shows the bioaccumulations rate of metals in the soft tissue of fish species and determined by using the modified versions of the formula. And also the bioaccumulation factors are determined by comparing the metal concentrations in the fish tissue and the metals of the concentration in the surrounding water media, which is mentioned in the units of the microgram per liter. This slide shows the statistical analysis that will be done for in order to get the results. Lastly, that's all from me, and thank you for your cooperation. Assalamualaikum warahmatullahi wabarakatuh.